Water is essential for life, but in many parts of the world, water quality remains a growing concern. What if we told you that the solutions to purify water may be closer than you imagine? In nature itself? Welcome to the video, From Soil to Solution, using siderophores to empower plants in water filtration.
Klebsiella pneumoniae is a bacillus commonly found in the human gastrointestinal tract and in the environment, including water and soil. This bacteria can colonize the skin and mucous membranes, which facilitates its transmission in hospital environments. It is characterized by its ability to form biofilms, and it is resistant to disinfectants. Klebsiella pneumoniae belongs to the Enterobacteriaceae family and is described as a gram-negative, encapsulated and non-motile bacterium, and that appears as rod-shaped under the microscope. The size typically ranges from 0.3 from 1 micrometer width and 0.6 from 6 micrometer in length. The associated pathologies include pneumonia, which is a serious infection that often presents with dark brown or jelly red sputum, and it's more common in people with diabetes or alcohol use disorders. Bacteremia and sepsis. Klebsiella pneumoniae is a common agent in cases of bacteremia, where the bacteria enters the bloodstream, and sepsis is more common, especially in immunocompromised patients. Urinary tract infection. This bacteria is responsible for urinary infection, which can manifest as cystitis or pyelonephritis. This type of infection are especially common in patients with endocrine catheters. And for end, surgical wound infection. The bacteria can infect surgical wounds, especially in patients who have been exposed to invasive procedures or who have implanted medical devices.
The virulence factors of the Klebsiella pneumoniae are due to wide variety of elements that can cause infection and antibiotic resistance. Among the most important are the capsule, lipopolysaccharides, fimbriae, and siderophores. In this case, we will focus on siderophores. Klebsiella pneumoniae is capable of producing different types of siderophores, such as Yersinia bacteria and Aerobactin, which are mixed siderophores because they contain components from several families. It also produces Enterobactin and Salmonellin, which are catecholate type siderophores composed of three phenolic rings. Siderophores capture iron from the host, which facilitates the development of Klebsiella pneumoniae in each of the pathologies in which it is involved.
Enterobactin, with the highest known affinity for iron of any molecule, is a prototypic catecholate siderophore and effectively outcompetes host iron-binding proteins for iron. That molecule is synthesized for the anti-PCDEF operon, and those ferric siderophores are actively imported by bacteria through siderophore-specific outer membrane receptors that are dependent on the DOMP, XUB, XUD energy transducing system. The receptor has the same two domains, 22-stranded beta barrel and N-terminal plug, common to the TBDTs. Extracellular loops on the plug domain and the beta barrel are the nodes NL1 to NL3 and L1 to L11. PFEA has loops L3, L4, L7, and L10 that cover the extracellular phase of the structure. The ferric siderophore is linked to PFEA by residues of loops L2, L3, L4, L7, and L11 on the extracellular surface. A space-filling analysis of this extracellular phase reveals only a narrow tunnel with a diameter of approximately 3 Armstrong in the epistructure, and it is also seen in the structure of the FAN complex. Comparison of the structure of the FAN complex with the epistructure shows that L4 has undergone a large conformational change to interact with the enterobactin molecule. The change in glue 327 conformation of L4 results in the side chain reaching further into the pore to form a salt bridge with arch 100. Causing a peptide bond in Pro28 to be inverted, resulting in the formation of an anti-parallel beta sheet instead of the alpha helix and disorder of the DOM box, facilitating internalization of the siderophore.
So, what is the importance of the iron in humans? Well, the iron is a vital mineral for humans, participating in numerous metabolic processes. It is a key component of enzymes and molecular complexes, like hemoglobin, playing a crucial role in synthesizing DNA through the enzyme ribonucleotide reductase. Iron also facilitates electron transport due to its ability to accept and donate electrons. Moreover, iron is essential for nervous system function, as it involved in the synthesis, degradation, and storage of neurotransmitters. The distribution of neurotransmitters like GABA and dopamine closely aligns with iron levels, suggesting its a significant role in dopaminergic and GABAergic functions. However, excess iron can accumulate intracellularly as ferritin and hemosiderin in the liver, spleen, and bone marrow. An average healthy adult contains about 3 to 4 grams of body iron.
Environmental pollution is a significant problem in today's society. A study conducted by Singh in the city of Varanasi revealed that the continued use the wastewater from the Dinapur plant to irrigate vegetables has led to the accumulation of metals such as cadmium, lead, and nickel, exceeding safe levels for human consumption. The daily intake of these metals varying according to the type of vegetables. Fresh vegetables provide a higher amount to copper, lead, nickel, and chromium, while leafy vegetables are the main source of cadmium and zinc.
Hereditary hemochromatosis is an autosomal recessive disorder that disrupts the body's regulation of iron. It is the most common genetic disease among Caucasians, with men experiencing higher rates of iron overload than women. About 85 to 90% of those affected are homozygous for the HFI gene mutation C282Y. While many individuals may carry this mutation, only around 10% will show clinical manifestation, which can be nonspecific in early stages. Symptoms may include weakness, lethargy, arthralgias, and impotence. As the condition progresses, more severe complications can arise, such as cirrhosis, hepatocellular carcinoma, osteoporosis, diabetes, and can lead to restrictive cardiomyopathy and serious heart issues, including dysarrhythmias and heart failure. We propose using the ABCDEF operon from Klebsiella pneumoniae to create a recombinant vector system. This system aims to enhance siderophore production in plants, improving their ability to sequester iron from contaminated water sources, and employing a transportable platform designed for the cultivation of flowering plants. Thus, at the moment they are no longer needed, they can be removed from the platform and transported to another location that requires decontamination. What is our objective? Produce engineering plants. Utilize an ABCDEF operon from Klebsiella pneumoniae, reduce the amount of iron in contaminated water.
Now let's take a look at the materials we'll be using in this experiment. The materials can be divided into categories based on the step of the experiment, as you can see. So we have the bacteriolysis, PCR reaction, recombination reaction for entry vector and exit vector, and to create competent agrobacterium cells and to transform plants. So I'm not going to go into detail about every single material here, but don't worry, you can pause the video at any time to take note of them. So let's dive into the detailed methodology. Well, once we have identified the gene encoding enterobactin and its transporter DOMB in websites like NCBI, we need to extract the DNA from the bacteria. So we're gonna pick colonies from the agar plate and resuspending them in 100 microliters of molecular grade water in an Eppendorf tube. Next, we incubate the suspension at 100 degrees Celsius for about 10 minutes to lyse the bacterial cells. After that, we centrifuge the sample at over 1000 revolutions per minute for 10 minutes. And once it's done, we transfer the supernatant into a new tube. This liquid contains the nucleic acids we need for the next step, the PCR amplification. In this step, we'll use specific primers designed for our desired gene sequences, so we prepare the PCR reaction and be careful to mix everything together, and then place to the tube in a thermocycler. When it's finished, we need to run a 1% agarose gel electrophoresis to verify the size of our product. If it's correct, we have successfully amplified the correct sequences. So we prepare our PCR product with blown ends for cloning. We add the PCR product in the p-entered the topo vector to an Eppendorf tube and we incubate it. And now we have to do competent E. coli cells following the standard transformation protocol for the competent cells. Then plate them on selected media containing kanamycin. This helps us to select the E. coli cells that have successfully taken up the recombinant p-entered the topo vector with our insert. The next step is the LR recombination. Here, we transfer or insert from the p-entry vector into the PEG 301 destination vector, and for that we mix equal amounts of the entry clone and the destination vector in a suitable reaction buffer and let the LR clonase carry out the recombination. Once it's ready, we plate the mixture onto selected media containing the same antibiotic we used before and incubate overnight. Now it's time to transform Agrobacterium tumefaciens with the recombinant gateway destination vector, using either a heat shock or electroporation method, and then we plate them on selected media containing kanamycin 2 to select the successful transformants. and then incubate the plates overnight at 28 degrees Celsius. And now we are ready to move on to the plant transformation. First of all, we use young tissue from Iris rhodocurus and we sterilize the plant tissues by first soaking them in 70% ethanol, followed by the treatment with sodium hypochlorite, and after that, we rinse the tissue through with sterile water. Next, we inoculate the sterilized plant tissues with agrobacterium suspension in a co-cultivation medium, like MS medium, and let them incubate in the dark at room temperature for 2 or 3 days. And once the co-cultivation period is over, we transfer the plant tissues to a selection medium containing chloramphenicol to select for cells that have successfully integrated the CMR gene from the exit vector. After several weeks, we'll subculture the surviving tissues onto a regeneration medium, again, MS medium, but this time supplemented with growth regulators. And finally, we transfer the plants to soil in the platform and begin the process to acclimatizing them to their new environment, gradually adjusting them to inundation conditions. And now we have plants capable of removing heavy metal contamination from any aquatic environment. We're excited to see how these plants perform in real-world conditions and hope they will make a positive impact in our environment. Thank you for joining us on this journey.